Okay, today we are going to talk about dihybrid crosses, where we're looking at the inheritance of two different traits. So Mendel asked the question, does the gene that determines if a seed is round or wrinkled have anything to do with the gene for seed shape? Must a seed that is yellow also be round? And the answer is no. The traits are not connected to each other. They are inherited independently. So we have to treat them like that and kind of look at all the different possible combinations that can be inherited. So to test this idea, he crossed a plant that was round and yellow with another plant that was wrinkled and green. So he crossed a plant that was all homozygous dominant with a plant that was homozygous recessive for the two traits. Similar to what he did with his monohybrid cross, where he wanted to see kind of what came out with that F1 generation. It should be heterozygous. But what happens when you do it with two traits? So the genotypes of the two parents, we just kind of put them together. We are homozygous dominant for round and homozygous dominant for yellow, homozygous recessive for wrinkled, homozygous recessive for green. So we essentially have a true breeding cross, but with two traits instead of one. So we need to figure out, well, what are the possible combinations that can result? Because remember, each parent can only provide one allele. So how we do this is we kind of use what's called FOIL. So we take a look at the first parent genotype, the first allele of the first trait, and it'll pair up with the first allele of the second trait. So that is our first allele combination. Our second allele combination, first allele of the first one, second of the second one. And then we go second allele, first allele. And then finally, second and second. So 4 different combinations. And we do the same thing with the other one as well. So, first allele, first ones of each, first and second, second and first, and then second and second. So you have 4 different combinations that can happen with each parent genotype. So when we look at this, remember, we have to follow 2 laws. The law of segregation basically says that each parent can only give you one allele, which is why we only have a pairing of one R and one Y. And the law of independent assortment says that the different pairs of alleles segregate independently. So this is why we have to do the FOIL, where we have to see what are all the possible combinations that each parent can give you. So if we look right here, with our combinations, this would be in one gamete. This would be in another gamete. This would be another gamete. This would be another gamete. Let's say that's the parent. Let's say that's the father. And the mother, the egg cells have different possibilities as well. So we have to acknowledge these possibilities based off the law of independent assortment. So what we end up with here is 100% are going to be heterozygous. Similar to what he did with the monohybrid, he then crossed the hybrids, self-fertilized them. So in essence, with our F2 generation, we have parents that have heterozygous for both alleles. And now we have to do kind of the same thing. Since both parents are the same, they'll end up with the same different combinations. So first allele with the first allele. So we have capital R, capital Y. First allele, second, capital R, lowercase y. Second allele, first allele, lowercase r, capital Y. And then second allele, lowercase r, lowercase y. And since they are the exact same, we will have the same combinations on the other side as well. So because of this, we have 4 combinations instead of 2. We have a 16 square Punnett square. So what we're going to do is we're going to put our different combinations across the top. So let's say this is parent 1. So parent 1 we're going to put right here. So I have that combination. Oops, the first one was a capital Y. Sorry about that. So I have this combination. I have that combination. I have that combination. And then finally, the last one, kind of the recessive combination. And with parent 2, put that vertically. I have that going down. Same idea. 2 capitals. Capital lowercase. Lowercase capital. And then lowercase lowercase. Since I have 4 combinations of each parent, 16 squares, now I simply put them together. Make sure to put the same letters together to figure out if they are homozygous dominant, heterozygous, homozygous recessive. So my 2 R's right here are 2 capital and 2 uppercase. I have an uppercase R and I have another uppercase R and a lowercase y. Uppercase R, lowercase r, and another uppercase y. And you just keep going until you fill out the entire table with all the different possible combinations. And you typically always want to have your dominant alleles written before your recessive ones. Halfway there. And now we have a fully completed table. So now what we're going to do is we're going to go back and figure out our phenotypic results with our table. So round and yellow are going to be our dominant. So dominant for round, dominant for yellow. So I see that right here. 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 are going to be dominant and dominant. Now we're going to take a look at dominant for round, but recessive for color. So dominant, recessive. I see that right here. Dominant, recessive. 1, 2, 3. So 3 are right there. All right, our next combination is going to be recessive dominant. 1, 2, 3. We got 3 there. And then finally, we have both of them are recessive, and there's only one possibility with that. So whenever you have a heterozygous cross with dihybrid, we have that nice 1 or 9, 3, 3, 1 ratio. Similar to monohybrid, where we have that nice 1, 2, 1 ratio with genotypes, or the 3 to 1 ratio with phenotypes, we're only really worried about phenotype here because there's a lot of possible genotypes. So that 9, 3, 3, 1 phenotypic ratio with a dihybrid heterozygous cross. All different possible combinations. So when he found this F2 generation, having this 9-3-3-1, similar to what he did with the monohybrid cross, this supported the idea that traits are transmitted to offspring independently of one another. And this led to the law of independent assortment. Each gene is not tied to another one in this case. They are inherited independently, which allows us to have several different types of combinations and much more variety compared to a monohybrid cross. So that's why we have a 16 square Punnett square instead of 4. Okay, that is it for dihybrid cross. Just like before, the best way to help you with this is to practice. Now there is a little bit of a shortcut you can use with the product rule, which we'll kind of go over another day. But thanks for watching. If you have any questions, please ask.